Alright, we're still in Chapter 3 of Kubi, 7th edition, the structure of antibodies. This is very, 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 very important. Please read these next few pages in as much detail as you can, because it kind of builds on everything else we do for the rest of the semester. The structure of antibodies. Antibodies are proteins. They're peptides. They're made up of 4 strands, so they're a tetrapeptide. They have 2 real light strands and 2 longer strands, which would be heavier strands. You have constant regions that don't change much between different antibodies that's found in a particular individual. But you have variable regions that change a great deal because those variable regions allow the antibody to bind to different receptors. There's some common features to the way these things fold up. You get a lot of beta-pleated sheet arrangements, and you get a lot of variability in the V regions of the domains. So read through these pages 80 and 81 definitely in a lot of detail, and kind of make sure you know where the heavy and light chains are. Put a big O star on page 82 and 83 because this is where they found out the different regions and the function of the different regions of antibodies. So at the bottom of 83, figure 2 is incredible. It's talking about the Fab region. Absolutely fabulous. The Fab region contains both heavy and light chain regions that are highly variable, that can be modified, and the more they're modified, they learn to recognize things that are have not been seen before in that particular human body or in that particular animal. So this is what binds to the antigen, the thing that's going to regulate or stimulate an immune response. Where if you cut the bottom half of the antibodies off and you just have the 2 heavy chains that are usually constant regions, then that falls out when cold. It's called the FC region. What happened is some British scientists discovered this. They went to the pub, they came back, and because they had put their propane digestion in the refrigerator, the FC region fell out of solution, so it fell out when cold. That's why it's called the FC region. So if you look at it like the Fab region is going to bind to the antigen receptor, that causes the FC region to kind of change its conformation, to stick its butt out. And that's going to cause all sorts of things to happen. That FC region is the activator, the effector region. And you need to find out as we're reading through this what the effector functions of antibodies are. So I can foresee a question on the exam that asks you to identify the effector functions of an FC region on an antibody that's bound to its antigen. That's a great receptor, or that's a great question for an exam. Now, if you look on page 84, there's a lot of different immune receptors that are found on the surface, and there's the M8C molecules that we'll discuss in a lot of detail later. You'll notice that the class 1 molecule has a beta-2 microglobulin, so it's got one long receptor with this beta-2 microglobulin is almost like a pegged leg that's under there, where class 2 is 2 separate branches that come together and can separate to bind to specific receptors. What I want you to pay close attention to is the C receptor side that's on the inside of the cytoplasm of the cell. That's going to be really short in some of these receptors, and it caused a lot of people to scratch their heads and go, wait a minute, these don't have enough C-terminal regions inside the cytoplasm to stimulate a signal transduction mechanism. But then you look at some of the accessory proteins that are down on the bottom part of that figure 319, and they have much longer tails that are sticking down in there. There's ICAMs, VCAMs, CD64s, there's CD8, CD4s. All of these kind of accessory proteins do have C-terminal regions in the cytoplasm that can stimulate a signal transduction mechanism. So it's like an extra control. It prevents something binding to the MHC1 or 2 or to the immunoglobulin receptors and sending a signal transduction without the cooperation of some accessory protein. So it prevents that autoimmune type dysfunction that can occur. Read through all the way through page 85, where it's talking about two major classes of antibody light chains. Then the next audio will be talking about 5 major classes of antibodies.